Ha, that
Perfect.
works? Okay, great. So reproducible brain charts, as mentioned, is an open data resource for mapping brain development and its associations with mental health. It's a project that was led by Golia Shafiei, who's a postdoc working with Ted Satterthwaite at the University of Pennsylvania. And one of the reasons that I put in all this, like the list of authors on the paper that came out and was published last year in Neuron, is just to give you a sense for how wide-ranging this project is, and includes many different institutions, really all over the world. And so here is kind of some of the institutions involved in collecting this data, and it's just really a masterpiece of coordinating multiple groups to do something together. The goal of the project broadly was to create a resource that people can use in order to study development, in order to study child development, adolescent development, and particularly to study brain health. So in the context of particular mental health. Um, Version 1.0 of RBC that's currently accessible includes data from 6,346 participants, ages 5 to 85. And though the age range is very wide, most of those participants are actually in the 5 to 22 age range. It takes its data from 5 major studies of brain development in North America, China, and Brazil. Those are the Brazil high-risk cohort, the Developmental Chinese Color Nest Project, the Healthy Brain Network, Nathan Klein Institute, which is the one that actually includes most of the participants that are above 25, let's say, are in that one data set, Nathan Klein Institute data set. And then finally, the Philadelphia Neurodevelopmental Cohort. So these are the 5 studies, and they've aggregated together the data from these studies and made those accessible through kind of a uniform interface. So what does this include? This includes measurements of both structural and functional neuroimaging at this point, as well as assessments of psychopathology. So different studies did their assessments of psychopathology in different ways, but as part of the project, the data were harmonized using what's called the bifactor model, so that every participant's level of psychopathology using these various different questionnaires can be summarized using one number, which is a p-factor. A low p-factor means low psychopathology, and a high p-factor means high psychopathology. If I remember correctly, the number is between 0 and 1. So it's kind of like one number that tells you something about psychopathology broadly. And then the data were processed. So the functional neuroimaging data were, all the pre-processing that you need to do with functional MRI data was done with those data. And then the structural data were processed using standard pipelines, FreeSurfer, and so on. And then the data were harmonized. One of the challenges with putting together a data set like this is that the data in every site were collected on different instruments, different scanners. And that means that they're not scaled relative to each other. You can see this here in this plot from the reproducible brain charts paper, where you can see that cortical thickness, for example, we know that cortical thickness decreases with age in the age range between 5 and 25. But you can see that each one of these studies had sort of different mean cortical thickness. And that's probably an effect of the instruments, not the differences between the samples. And so you can statistically correct for that using harmonization methods. And now the lines are kind of overlapping with each other. So everything was harmonized in that way. And then finally, everything is provided in what you might call data science ready formats. So these have all been processed into tabular data that we can then start doing all kinds of data science with. How can it be accessed? It's accessible through this website, reprobrainchart.github.io, where there's also descriptions of these different data sets and a little bit more information about how the data was processed. And you can download it using a system called DataLab. Within the website, there's a web page specifically about how to get the data, or it can actually be downloaded directly from AWS S3. We'll discuss a little bit more about how you can get data of this sort from AWS S3 on Friday when we do the data showcase, which will show you many different data sets that you can use in week 2 of NeuroHackademy. But today you'll actually be able to access that through the hub, assuming we figure out all the technical issues that we've been having with the hub. But there are, if you're interested in the future to kind of explore this data, there are different ways that you can access the data. Okay, and that was my bit about RVC. I'll stop sharing and kick it back to Noah.
Okay, thank you. Okay, so what I was planning to do next was to have all of us walk through this together on the hub, but since the hub is not working for many of us at the moment, that's obviously not going to work. I'm just going to kind of walk through this and follow along as best you can. The good news is that we've done a lot of the work for you here. So ignore this big pink banner. That's not our problem. What I've got here, oh, sorry. People on Zoom probably can't see my screen. 1 second. Okay. Let me see if I can hide this. Okay. Okay, so I'm here at the GitHub repository for the week 1 projects. Now, we've got this in the GitHub repository NeuroHackademy 2026/NH2026-week1-project. I've also posted this link in the Slack in the week 1 project whole channel. It should be at the bottom. Now, this repository here is you're not going to edit this repository directly, even though it is in the NeuroHackademy 2026 space. What we want you to do is we want your group, and probably each of you individually, to make a fork of this repository. If you don't know what a fork is, we will talk about that in the next session on Git and GitHub. And if you, just real quick, I'll say that if you have not yet configured your Git and GitHub on the hub, which probably most of you have not done, in your docs directory or in the shared/docs directory, there's a git setup.html file here that has links to where you can find information on how to set up Git. That said, if you aren't sure how to set up Git or if you've never done this before, come to the introduction to Git session and we'll do it all together. And that'll be after lunch today. Okay. So just to kind of walk through this right now. So I'm here at the NH 2026 week 1 project GitHub repository. I'm going to go ahead and click this fork button so that I can make a fork of the repository. And you notice that I'm going to fork it into my own account here. So I'm going to make a copy of this repository that is now owned by me instead of NeuroHackademy 2026. So I just click that create fork button. This will take just a minute, but you'll notice that it's already gone to Noah Benson /NH 2026 week 1 project instead of NeuroHackademy 2026. So I now have a fork of this project that is mine. And if I edit this fork, I won't edit the original template repository. So I'm now going to go over to Jupyter Hub. Again, my Jupyter Hub is still working. I'm not sure why, but it is still working. And I'm going to go ahead and open, using the launcher, I'm going to go ahead and open a terminal. This is just a terminal on the computer that's running your Jupyter Hub server here. So this is just like any bash terminal. And I'm just going to git clone git at github.com : and then I'm going to put my username, Noah Benson /NH 2026-week 1 project. Now, if you haven't configured your Git yet, this command will fail because it won't be able to authenticate with Git. And again, if you want help with that, we'll walk through how to fix this in the intro to Git session after lunch. But for me, I've already configured my Git account, so it's going to go ahead and just work fine here. Okay, and then you can see that it's created a copy of the repo here. So I'm going to go ahead. I'm going to make this just a little bigger for the people online. I'm going to go ahead and navigate into the GitHub repository. And you'll notice that there's an analysis.ipynb file here, and a Jupyter notebook. So this repository has been set up as if you were going to write a Python library into it. You don't have to do that for this project. We just kind of set that up as more out of habit. It's fine if you just want to do this project in notebooks, or if you want to write a companion library for the project, that's up to you. But what Ariel and I did was we went ahead and wrote an implementation of this project. We wrote our own model for this project. Okay, let me pause here and tell you what we're going to ask you to actually do in this project. The RBC data set has a whole lot of data in it. We aren't asking you to look at all of the data. We're going to ask you to look at just a subset of the data, which is the Philadelphia neurodevelopment cohort. So that is abbreviated PNC throughout the reprobrain charts data set. And specifically, Ariel mentioned this P factor or psychopathology score. And what we're going to ask you to do is to predict the P factor of a set of subjects from the Philadelphia neurodevelopment cohort of this data set by using some kind of machine learning model or method. And what we're going to do is we're going to give you a set of about 80% of the subjects, and we'll give you the P factor scores for those subjects. And then we're going to have a separate set of subjects that are our test subjects, where we'll give you all the information about them except the P factor. And your job will be to predict the P factor in those test subjects, and we'll score you based on how well you predict that P factor. So just to kind of restate that, we're giving you a training data set, which is 80% of the subjects, all of their data, including the P factor, for you to train your model with. And then we're going to give you test subjects, which is about 20% of the subjects, the rest of the subjects, who will not have the P factor included, and your job is to predict that P factor. Does that do people have questions about the kind of goal here before I keep going? Okay, great. Okay, so we've gone ahead and written some code here that will help you get started. And this notebook contains our very simple implementation of this project. So what Ariel and I decided to do was pretty much the simplest model we could come up with. We took the size of Brodmann area A, or Brodmann area 1, for all of the subjects, and we trained a linear regression using Brodmann area 1 to predict the P factor. Now, this is not a very good model. There's no real reason for me to suspect that Brodmann area 1 would have any correlation with a subject psychopathology score. So I'm not going to tell you right now how much variance we explain in the data sets, but it's not very much. This will kind of be a baseline that you can compare against when we get to actually looking at the results. Okay, this notebook here, this analysis notebook, my suggestion is that you start with this and modify it. It's got a lot of text and documentation in it so that you can understand it. If you have questions about it, feel free to message us. We're going to give you lots of time throughout the next few days to work on this in groups. But what I'm going to do right now is just kind of walk through it all together. So we've included a special library that we wrote this library. This isn't like a public library called RBClib that is specifically for accessing the data in this project. This is just to simplify things. And like Ariel said, we'll talk more about how to access data from S3 and such on Friday. But this library basically includes a type called RBC path, which is broadly similar to the path type from Python's pathlib. Long story short, for those of you who haven't used the path and pathlib types, path is just a way that you can represent paths on the file system. So like if you want to represent your home directory, and here I'll just as an example, I'm going to go ahead and run that cell. You can ignore that future warning. And then I'm going to say path.home. And you'll see that it says, your home is a POSIX path that's /home/Jovian. Okay, if I want to look in this directory, this NH 2026 week 1 project directory, I can put a slash, just like slash is used. I mean, obviously this is usually a division symbol in Python, but the path type overloads that division symbol to be a directory separator. So just like you separate directories with forward slashes on the command line, you can separate them with forward slashes using these paths, and I can say NH 2026 week 1 project. And that will represent this subdirectory of my home directory. And I can wrap this in parentheses and then say, for example, well, okay. I can iterate over this directory. So for example, if I ask for a list of iterating over the directory, it's going to give me all the subdirectories of that week 1 project directory. if you're not familiar with this, there's lots of help on it, and we're happy to help you with it. But the basic idea is it's a way to represent paths in Python. And this rbclib library implements a path that represents the RBC paths up in the cloud so that you can pretend like they're local. Okay, if that's not clear, I'm hoping it'll become clear as we walk through the example. Okay, so each of these subsets of the data set, and in particular, like I said, we're just focusing on the Philadelphia neurodevelopment cohort, PNC. They have multiple different kinds of data that have been processed. There's there's PNC underscore free surfer, which contains structural data. That's what we're going to focus on here in this notebook. There's PNC bids, which contains it's a bids directory, brain imaging data structure directory that contains all the MRI, the raw MRI scans from these experiments. And then there's some processed functional MRI data. Our recommendation here is that you focus on the PNC free surfer data. We're mostly recommending that because it's already processed. You can start working with it pretty quickly. It's fairly easy to understand. And our goal here with this project, again, is to get you to kind of practice with these tools, not necessarily for you to have like the most perfect project or most perfect model. You are welcome to use these other data subsets of the data set. They will just be more raw and more difficult to use. But we don't want to restrict how you answer this question. We're just going to show you in this notebook specifically how to use the free surfer derivatives. Okay, so in this cell here, I start by naming a subject ID just as an example subject. This is a subject you can look up from the data set, but this is just an example subject. And then I declare an RBC path object. You notice it starts with RBC : / and then the subset of the data set I want to work with, here PNC free surfer. And then the rest of this is just a path to the data. They have the data sitting in S3 and up on GitHub. And so this is just a path to the subject. And for the free surfer data, this is the path for every subject. So for any given subject ID, it's always going to be PNC free surfer / free surfer / sub - subject ID. And then here I'm just making sure that this is a directory. If this isn't a directory, we'll get an error. And then it's just going to print out all of the files that are in that directory. So I'm just going to go ahead and run this. And you see that it prints out a bunch of RBC paths with files that are in this directory. So for example, this subject's brain measures .tsv, which stands for tap separated value. So all the data lives in these files, basically. Okay, so if we want to read in one of these files, we can specify its path here. I'm pulling out specifically the region surface stats.tsv, which contains information about the sizes of various anatomical areas in the cortex for each subject. And here I'm just going to print out that we're loading the file and then ask Pandas to read it in as a CSV file. TSV files are just CSV files where you use tabs instead of commas, and that's why that works. And then it's just going to show the data at the bottom. So now this will take just a second to run because it needs to go load that data in. But here we have the results of this cell, the results of loading in this particular data. And you can see that there's various columns here that represent different surface area measurements on cortex.
And those are standard free surfer outcomes?
These are not quite standard free surfer. You wouldn't find these in a typical free surfer subject directory. These are summaries of them. So it's not. I believe you can find the free surfer directories in the bids, but this particular set of data is summaries of the free surfer. Okay, so this shows us that we can read in data from a single subject. But for this project, you need a few things, not just the ability to read single subjects. You first of all need to know who's in our training data set and who's in our test data set. We've gone ahead and put in the shared directory. There's a shared /data /RBC directory, and it contains two TSV files, one with the training participants and one with the test participants. So again, you're going to be reading in the training participants. That TSV file has a p-factor column. The test participants, on the other hand, does not have a p-factor column because that's what you're predicting. Okay, so this cell is just going to read in these TSV files, and then it's going to concatenate the training and the test data and just print it at the bottom. So that goes pretty quick because those are just a couple TSV files we're reading off of the drive. So you can see that there's some information, some basic demographic information here, like age and sex and handedness and such. And you can use these as part of your predictors as well. And if we go all the way over to the end, you'll see that there's a p-factor column. And for some of these subjects, all the training subjects, there is a p-factor value. And for the test subjects, it is all nan, meaning not a number. So again, this column, these nans are what you're going to be predicting. Okay. So there's a lot of text here. I'm going to go over it pretty quickly, but the idea is if you have questions about what these cells are doing, you can come and read it later. Here, I've just written a function called load fs data. You're welcome to use this, but it stands for load free surfer data. You give it a participant ID, and there's an optional argument here for a cache directory. If you don't change that optional argument, it will use a cache directory that has already been populated, so you don't have to wait quite as long to load all the data in. There's a lot of data here, though, so even with a populated cache directory, it still takes many minutes to load all the data in. Just FYI, I'm going to suggest that when you start working with this data, you load in a few subjects and use them as kind of a test set. And then once you've decided or have a bit of an idea what you want to try, then load all this data in, because it will take a little bit to load the data in. So what this function does is it makes your cache dir if you don't have a cache directory already, but you shouldn't have to worry about this if you use the default argument. It then creates an RBC path for the free surfer data for that subject, and it loads in that region surface stats.tsv file and returns the data frame. So all this does is load in that data frame we just saw for an arbitrary subject. So for example, we can pick out an arbitrary subject and just run this function, and I'll just show you what this does. It's pretty much what we just saw. It loads in this example subject's free surfer data and shows us the data frame of all those data. Okay, so I mentioned that Ariel and I came up with a model that just looks at Brodmann area 1. So I'm just going to start by taking this example subject's data and pulling out Brodmann area 1. So we look for anywhere where the structure name, for which is one of the columns in this data frame, is equal to Brodmann.1. And when we print out that particular row, we see that we have two of them, one for the left hemisphere and one for the right hemisphere, which is sensible. And we have things like the surface area, which is in square millimeters here. Okay, so we can extract the surface area by just extracting those particular rows we pulled out. And then we're just going to add them together because, like I said, our model is very simple. We're actually going to use the bilateral Brodmann area, not the left or the right, or the left and the right separately. So we're just going to sum those. And that gives us the bilateral surface area of Brodmann area 1 for this particular subject. Okay, so now that we've established that we can do that in a subject, here's a little function that does that in some arbitrary subject that we provide. So you can pass this load BA1 surface area a participant ID, and it will do what this cell up here did. It will read in that subject's data and find the Brodmann area 1 labels and give you back the bilateral surface area of that subject's Brodmann area 1. And just to test it here, we can use our example participant ID and make sure that it loads in correctly, and that works. Okay. So this next cell is sort of the cell that does all the work. You'll notice here that there's a little bit of like printing some stuff and setting up some variables. This makes a little display bar so that we can watch the progress, because loading in all of the subjects does take quite a while. But basically what this does is it goes through our data frame of all of the subjects, and for each one, it calls this load BA1 surface area function that I just wrote, or just ran. If there's an error, because some subjects have missing data, so there are sometimes errors, it just puts in a nan. Otherwise, it creates, or it adds to our data frame. It creates a new data frame called all vars here, and it adds to the data frame the participant ID and the surface area for Brodmann 1, and the p-factor if this subject has a p-factor. Okay, I'm going to start running this cell and you'll see that well, you'll see. So you notice how quickly it's ticking forward, if you can even see that little stripe of blue. That every time it changes, it's loaded a subject. There's a lot of subjects. So even though this is loading off of a pre-populated cache, it takes about 20 minutes or something to load all the subjects. So that just gives you an idea of how you're going to have to run these analyses. You're not going to be able to, if you're coming up with lots of ideas and you want to run them all, you're not going to be able to just run them in a minute. You're going to have to take a little time to actually test it on all the subjects. Okay, I'm going to leave that running, and I'll just explain what the rest of this does. Basically, so this builds up a data frame of all the subjects that has just the participant ID, the Brodmann area surface, Brodmann area 1 surface area, which is what we're using as our predictor, and the p-factor, which is what we're predicting. So we then use the scikit-learn linear regression type. And if you're not familiar with scikit-learn, there'll be more information about this tomorrow in the machine learning session. But basically, we're going to do a very simple linear regression. We create a matrix by pulling out the thing we're using as to predict our p-factor and the thing we're predicting, which is the p-factor. So what we do here is pull out basically a 2-column, many rows matrix of measurements for each subject. We pull out just the rows where there are no NANs, because NANs indicate that a subject is part of the test data set because we don't have a p-factor for them. And then we train our linear regression model using the training data, the training subjects. Now I'll go ahead and cue this to run, but it's going to take too long. This is going to still be loading when I finish here. So you'll just have to believe me that this works when it runs. So once we've trained this linear regression model, or fit this linear regression model using our training data, we can come down here and pull out just the BA1 surface data for our test subjects by pulling it out of these test variables. And then we can apply our linear regression model by telling it to predict based on the Brodmann area 1 of our test subjects. And that will give us predictions based on the linear regression for each of the test subjects, a prediction of the p-factor for each of the test subjects. Now, again, this is a very simple model and not a very good one. So we expect this to do pretty poorly. And what these last couple cells have are just instructions on how to save your results. Don't worry about that right now. We will talk about how to save your results at the end of the week on Friday before you submit these. So for now, if you can calculate the results, that's all you need to worry about. Okay, so we encourage you to we've allocated quite a bit of time for you to work on these projects in groups. You've got time right now. In fact, what we're going to do in a minute is release you all to your groups. We've already set up owls in all of the rooms. So if your group is Alder 107, you go to Alder 107 and there'll be an owl in there. For those of you online, if your group is Alder 107, go to the Alder 107 Zoom room, and you'll meet your group there. For those of you who are in person, please do not forget that you have online group members. There are more of you than there are online people. It is just very easy to miss communications or to not realize that they're talking and they're trying to get in touch with you. Please don't forget your online participants. We encourage you, like I said, we encourage you to be creative in how you try to solve this. The one thing I'm going to ask you not to do, this is a public data set, so all the answers are out there if you go look hard enough. Don't just go look at the answers. That's my one request here. Otherwise, you're welcome to go look at the bids data, look at the process data, and try to use it. I will warn you that will be a little bit more work and may take a lot of time, and you may find that you don't have time to do anything very fancy. That's kind of by design. We want you to focus a little more on using the tools and kind of getting used to working in groups and stuff, not necessarily on going crazy with the analysis just yet. Okay. Any questions broadly about the project before we release everyone? Yeah.
Do we just assume that linear regression is the best way to go about the data?
Oh, I would not assume that. So, yeah, good question. We use a linear regression model here because linear regression is just a very simple, easy to understand model. And also because we wanted to set a fairly low bar for you all. Not that we don't have faith in you. We just, we didn't want to pick a model that accidentally did really well. So we picked a very simple model, linear regression. My advice would be to try at least one or two different models. Probably not. I mean, linear regression is a great place to start for any project, pretty much, or any kind of prediction project like this. It's a good baseline, and I would potentially encourage you to try linear regression. Just don't expect it to be the best model. And if you're not sure what kind of models to try, scikit-learn is a great resource. It's full of models that are about as easy to use as this linear regression model. If you want to do a random forest, it's pretty much the same thing. You just replace linear regression with random forest. Scikit-learn is pretty good for those kinds of models, so I'd suggest looking there. Other questions? If anyone online has a question, just shout it out. Okay. Oh yeah.
Do we have chance to get a score of testing based likely for the timeline?
So we will not give you the scores for the test subjects until after you've turned in your estimates, and that's because we don't want you optimizing against the tests data. So obviously, if we give you the scores, you can come up with some completely arbitrary, crazy algorithm that produces exactly those scores. That's not interesting. What we want to know is if you're able to look at this data set and train it to produce some good answers. And I'll just say this one more time. What this notebook here does, and what we're kind of asking you to do about in terms of looking at the free surfer data in particular, is kind of like modern day phrenology. Like we're asking you to look at the sizes of anatomical areas of the brain and try to predict someone's psychopathology score. We're not expecting you to like get a really high score here. If you can explain 30% of the variance in this data set, you're doing awesome. So don't go at this expecting that like by looking at the sizes of brain areas, you're going to explain all the variance. That's not the point of this project. That said, groups have surprised us in the past. So like I said, be creative. You may find methods that work really well. Okay, any final questions?
What if we can't access Jupyter Hub?
So yeah, good question. If you can't access Jupyter Hub right now, what I'm going to ask you to do is go talk to your group, meet your group, maybe talk about different methods you'll use. We are going to try to get this fixed before the end of lunch. Fingers crossed. If the Jupyter Hub is still not working at the end of the day, we're going to have to brainstorm about something else we do. But my assumption is that by the end of lunch, this will be fixed. For now, basically, you've got until the end of lunch until the next session. Like the rest of the time is just for all of you to work on these projects. And you'll notice in the schedule that there are a few other times that we've allocated for you to work on these projects. And anytime you want to find, that's great. So for now, I'm hoping that you can all kind of do some planning and talk about models and that kind of thing, or ideas for analysis. If any of you can access the Jupyter Hub, great, you can get started. Again, we apologize that it's not working, but we'll hopefully get it fixed soon. Yeah.
One was immediately download the paper, but the answer's kind of in there, right?
So yeah, if you read the paper, you're not going to find a list of the p factors. And the p factors are out there, but they're not easy. Like this is just my impression is that I had to look pretty hard to find the p factors. So if you're just kind of poking around, you're not going to accidentally run into them. They are out there, but my ask is that you not go try to find them. Okay. If you have other questions, feel free to put them in the week one project old channel. If you sent me an Ariel message about not having your GitHub username in the list, we'll assign you a group pretty soon. Otherwise, yeah, go find your spaces. If you're in the Elder Auditorium, you can come down here. You'll be using this owl. All right. We'll get started and we'll see you all at and after lunch.